Thank you. Thank you for the introduction as well. My name's Mira. I'm one of the orthopaedic registrars at Norfolk and Norwich Hospital, and I did this research at the University of East Anglia. I don't have any conflicts of interest. So we know that Dupuytren's pathogenesis involves a complex interplay between genetic and environmental factors, and there's a constant dialogue between cells and the extracellular environment. We know that matrix stiffness influences phenotypic differentiation, fibroblasts exist along an activation spectrum, and numerous myofibroblast induction pathways exist. We also know that Dupuytren's nodule-derived fibroblasts demonstrate extended and significant contractile forces. And there's multiple disease-associated SNPs that have been identified. A small number of these SNPs result in amino acid sequence alterations termed non-synonymous SNPs, and these SNPs have a high potential to influence protein function. We know that Dupuytren's is a complex disease, and complex diseases have small contributions from multiple genes, and multiple factors that contribute to the disease progression and outcomes. However, SNPs may have synergistic biological effects, and in particular, non-synonymous SNPs may have strong biological significance and present potential therapeutic targets. One such non-synonymous SNP is this SNP, which is in the MMP14 gene. It's a G to A base substitution, resulting in an amino acid change. And we know that variants have previously been identified to have reduced cell surface collagen-electric activity and a reduced ability to activate pro-MMP2. However, little is known about this SNP's role in mechanotransduction. We do know that blockade of MMP14 and MMP2 using small interfering RNAs does reduce contraction of nodule-derived fibroblasts on fibroblast-populated collagen lattices, but beyond that, we don't really know what the role of MMP14 may be. Furthermore, the MMP14 protein may influence numerous pathways involved in Dupuytren's through its substrates, and you can see that there's a whole range of different functions of the MMP14 substrates, including ECM remodeling, activation of the wider proteolytic cascade, activation of pro-fibrotic pathways, and regulation of cell motility and invasion, as well as immunomodulation. We also know that the MMP14 gene and also MMP14 are involved in Dupuytren's through the work of single-cell RNA sequencing, which shows that MMP14 expression is the highest in the most active subpopulation of fibroblasts, along with other pro-fibrotic genes. And increased relative expression of MMP14 mRNA in Dupuytren's nodules is correlated with clinical recurrence after surgery. So we have this feedback loop where increasing tension in the extracellular matrix is a key driver of fibroblasts in myofibroblast differentiation, but also fibroblasts can influence extracellular matrix stiffness and composition, and an abnormal extracellular environment further drives the profibrotic cell phenotype. So we have a chicken and egg situation. What came first, the chicken or the egg, or the abnormal cell, the abnormal matrix, or the abnormal response of a normal cell to normal matrix, resulting in abnormal cells and abnormal matrix. So the aim of this study was to compare the response of nodule-derived fibroblasts from patients of different MMP14 genotypes with increasing matrix stiffness. So we had 3 genotype groups, homozygous, heterozygous, and the wild type, and also extracellular stiffnesses of 2, 12, and 72 kilopascals. We received tissue donated by patients undergoing primary surgery, and we genotyped our fibroblasts from nodules for the MMP14 SNP, as well as the 25 additional SNPs that were previously identified in the genome-wide association study. We designed primers using PrimerBlast, and then also genotypes for all of the 26 SNPs using a custom panel, which was validated by Sanger sequencing. We had nodule-derived fibroblasts, which were seeded in hydrogels. These are collagen-coated hydrogels, and these were incubated overnight. Cells were fixed, permeabilized, and stained for filamentous actin. And we then imaged and analyzed the images. We had 3 experimental repeats per patient, and then within that, each experimental repeat had 3 biological repeats. So patients in each MMP14 group were matched according to age, gender, and clinical features, and there were no statistically significant differences between the MMP14 genotype groups for age, smoking, alcohol, number of operations, and number of digits operated. This is just a summary of the demographics within our study, and you can see that they're classic Dupuytren's patients with a strong male predominance, older age, and a mixture of different risk factors. However, overall, there wasn't any significance between our genotype groups. Additionally, we were able to generate a SNP score through genotyping for the other 25 SNPs, so a total of 26 SNPs. And we were able to code this with additive coding, whereby having a wild type scored you 0, heterozygous 1, and homozygous 2, or dominant coding, whereby if you had wild type SNP, that was 0, and either homozygous or heterozygous scored you a 1. There was no statistically significant difference between our genotype groups for the SNP scoring. And this is just a very busy slide showing that we were able to generate this data for our 9 patients, so 3 in each genotype group for our 26 SNPs. And you can see that there's a difference in the scores in terms of how they were scored. However, the overall, there wasn't any difference in clinical significance. And this is just a summary with the p-values for our demographics and our genotype groups. So what did we find? So this is a representative image of cell area. On the top row, you can see that this is our wild type fibroblasts, whereby you have an increase in cell area with increase in gel stiffness. So at the 2 kilopascal gel, so top left, you can see that there's smaller cells, and then this progressively increases with our increasing stiffness, which is what we would expect. On the bottom, you can see that this is our homozygous SNP, so AA, and you can see that these cells appear to be slightly larger at the 2 kilopascal, and they remain large at the end, so they seem to have a lesser effect. And this is our summary slide. So you can see that this is mean cell area of our different genotype groups. In the dark grey, we've got the wild type, and then medium grey, heterozygous and homozygous variant, with increasing gel stiffnesses of 2, 12, and 72. And you can see that within the wild type group, there's a statistically significant difference in the mean cell area of fibroblasts from patients on 2 kilopascal and 72 kilopascal gels. However, this statistical significance is lost within the heterozygous and homozygous group. And you can see that overall, our starting point, particularly in the homozygous set, is much higher in terms of the 2 kilopascal cell area. So what does this mean? So we can see that wild-type fibroblasts show the appropriate response to increasing extracellular matrix stiffness, and this is an appropriate response to outside-in and inside-out signaling. However, there is a loss of appropriate response within the heterozygous and homozygous genotype groups. And there's a number of potential mechanisms, whether it's aberrant recognition of the extracellular matrix or an already activated phenotype with large cell area, which is minimally affected by the extracellular stiffness. So this study offers an insight into the mechanotransduction effect of this non-synonymous SNP. We know that the wild-type fibroblasts have an appropriate response. However, the heterozygous and homozygous fibroblasts have lost this response. And further work is needed in terms of increasing our numbers so that we can look at this in more depth, and also looking at our combinations of SNPs and our multiple genetic risk scores. Thank you. These are just references if anyone.
Questions from Mira? Yes, Dominic.
Thanks, Mira. Could I just ask, do you know what the stiffness of a Dupuytren's nodule and a Dupuytren's cord is? I may have missed this earlier, but I've seen these variations, and I wonder what we find in the hand.
Yeah, so there's a difference in the reported cord and nodule stiffnesses, and also there's something called the transitional zone, which is that kind of intermediate from when nodule becomes cord. I think cord is very much at the upper end of the hydrogels that we tested, but I think within the hydrogels, the reason why it's kind of a simplistic model and why it's very helpful is because it just allows us to look at the response rather than an absolute number. And you can imagine that different patients have different sort of stiffnesses of their cords and nodules. So we're looking at more of a behavioral element of these cells rather than an absolute number of cord and nodule.
Any other questions? Okay, thank you very much.